Hello everyone and welcome to the third video for Life on Earth. You should now know all about biotic factors and you should be able to pause the video and answer the starter question on keys before I go over the answer. So the answer is that if we look for the organism, first of all, so this part of the question here, we need to find an organism that has no spots on its body, and then we have to go to statement 3. So fewer than 12 legs. We have to go to statement 2, so it has legs. And the only one that is that would be your spider. So spider would be your first answer. So well done if you managed to get that correct. And now that we've got the statement that leads to 2 of the answers, we can now go back to this part here, and we now know that it's going to go to statement 4. Then we need to fill out the opposite of shell. So the opposite of shell would be that it has no shell. And then finally you need to figure out what this last organism is going to be. So it's going to have no shell, and then we had to go to 4, so it'll be no legs. So that would be your earthworm. Well done if you managed to get that correct. If not, you need to go watch the other video again. So the other part of the question says give 3 features of the beetle mentioned in the key. So to do that, we need to go to the beetle, which is here, and then it says it's got spots on its body, so that would be 1 feature. And then it says that we've went to 3, so we've got to find the go to 3. And then it's got fewer than 12 legs, so that would be another feature of it. And then you've got to go to 2, so go to 2 would be that it has legs. So its 3 features would be that it's got spots on its body, it's got fewer than 12 legs, and it's got jointed legs. So well done if you managed to get that correct. If not, then you need to go back and watch the last video again. Today we're going to be moving on to abiotic factors, which is still part of key area 2, distribution of organisms. If you want the worksheets to go with the video, remember to pause the video and go to Free Life on Earth, 2, distribution of organisms, 2, consumables and activities, and a PowerPoint is found in PowerPoints and is called 2, distribution of organisms. So today we're going to be learning about abiotic factors and sampling and indicators. So our learning intention is to state what abiotic factors are, to describe how abiotic factors affect biodiversity, describe the methods used to measure abiotic factors, to state the possible sources of error with each method and explain how to minimize them, to explain what a transit is, and to state that indicator species are species that by their presence or absence indicate environmental quality levels of pollution. To be successful, you will be able to tell the difference between biotic and abiotic factors. You'll be able to write down how abiotic factors affect biodiversity. You'll be able to create a table to summarize how you measure abiotic factor, which will include the possible sources of error and how you minimize them. You'll be able to write down what a transit is and describe how it can be used to show how abiotic factors affect biotic factors. And you'll be able to explain what indicator species are and give some examples. So can you remember what the terms abiotic factors and biodiversity mean? Pause the video and try to write out the answer before I go over it. So the answer is that biodiversity is the variety of different types of species present in an ecosystem, and an abiotic factor is a non-living factor which affects population numbers. So for example, this could be light intensity, soil moisture, pH and temperature. So abiotic factors affect biodiversity. Do you have any ideas of how abiotic factors might affect biodiversity? Pause the video and write down what you think before we go over the answers. An example where abiotic factors affect biodiversity would be soil pH. So different organisms can survive best in different pH levels. So for example, your bee orchid, your sheepspit, scabious, your rock rose and thyme and your land snail, they all survive in alkaline soils, whereas your rhododendron, panthecium, your linghever, your sundew and your butterwort, they all survive in acid soils. So obviously pH can then determine what organisms are going to live where. So what plants and animals are going to live where their let's look at another example, which would be temperature. Temperature can affect biodiversity because different organisms need different temperatures to survive as they're adapted to different conditions. So, for example, your Arctic fox and your conifers are adapted to cold conditions, so can only survive in cold conditions, whereas your desert fox and your cactus are adapted to hot conditions, so can only live in hot conditions. So, your temperature will affect the types of organisms that are there. Pause the video and write down how you think these abiotic factors will affect the daisies in the field before I go over the answers. So why will light intensity affect the numbers of daisies growing in a field? Why will temperature, and why will carbon dioxide levels? So the answers are that light intensity will increase the number of daisies because the daisies need light for photosynthesis to grow. Temperature will also increase the number of daisies because they need temperature to germinate. And carbon dioxide will increase the number of daisies because they need carbon dioxide for photosynthesis. So well done if you got that right. So light intensity, temperature, and carbon dioxide are all abiotic factors that will affect the biodiversity of plants such as daisies. And then that will affect other organisms. Number two abiotic factors that affect biodiversity are acid rain and global warming. Acid rain affects biodiversity because the pH of habitats are affected by acid rain. Acid rain is formed when sulfur dioxide and nitrogen dioxide are released when fossil fuels are burned. Acid rain indirectly decreases plant biodiversity as the acid destroys leaves so the plants can no longer photosynthesize. Acid rain decreases fish biodiversity and it lowers the pH of fresh water, which leads to fish deaths. Global warming also decreases biodiversity. So global warming does this because the temperature of habitats is affected by global warming. Global warming is caused when fossil fuels are burned and carbon dioxide, nitrous oxide, and water vapor are released and form a blanket around the Earth's atmosphere. These gases trap the heat and cause global warming. An increase in temperature decreases biodiversity as enzyme-controlled reactions do not work properly. The following natural disasters also decrease biodiversity. These include forest fires, earthquakes, volcanic activity, tsunamis, and wind. We already looked at why we need to investigate an ecosystem. We answered the question why we needed to find out what organisms live there, how many organisms, and why they live there. And it's because we rely on organisms for raw materials, food, and for medicines. So we need all of these different organisms. So it's important that we know what an ecosystem has in it. Now we're going to look at sampling abiotic factors. Can you remember what an abiotic factor is and give examples of it? Pause the video and try to write down what an abiotic factor is and what examples that there are before I go over the answer. So the answer is that an abiotic factor is a non-living factor that affects the distribution of organisms in a habitat. And examples include light intensity, temperature, pH, and soil moisture. So in third year, you should have measured some abiotic factors, and we're going to go over this in more depth. In third year, you should have used a light intensity meter. So you use a light intensity meter to measure light intensity. So first of all, you need to make sure you switch on the light intensity meter and set it to the light meter position. Then you need to make sure the meter is held so that the light sensitive panel is directed towards the light source to be measured and you don't cast a shadow over it. Then you need to take the reading from the light intensity scale once the pointer has stopped moving, and then all measurements are taken as near the same time as possible during a period of constant light intensity so that a comparison can be made at different locations unless you're doing it over time. So there's the two examples of light intensity meters, and you can see the top has the sensor on it. The errors that we can make when we are using a light intensity meter is that we might not switch it to the light meter setting, so you might not switch it to the right sensor. So to minimize that source of error, you need to check the settings and make sure it's switched to light and not soil readings. Another possible source of error that you might make when trying to do a light meter reading is casting a shadow over the sensor. To minimize this, make sure that you do not shade the sensor with your own shadow. So hold it in a way that the sensor is not shaded by your own shadow. And the last one is the time of day, so that could be another source of error doing it at a different time of day. So to minimize this error, you should take readings at the same time, since light varies so much over the day unless you're tracking changes over time. So well done if you managed to remember that all from third year. So let's move on to soil moisture. So you should have used the soil moisture meter in third year. So soil moisture is measured by a soil moisture meter. And again, you need to make sure that you switch the meter on the correct setting. So this time you'd want to set it to the soil moisture meter position. And then you need to insert the soil moisture probe. So you always have to talk about the probe, not the meter, or you will not get your marks. So your probes are these bits here. So the probe has to be pushed into the soil at an equal depth. So for example, 40 millimeters. The reading is then taken from the moisture meter scale once the pointer has stopped moving. So that's just this bit here. And then the probe is wiped with a tissue between samples to prevent moisture from one reading affecting the next one. So let's look at the possible sources of error that might happen when you're doing a soil moisture reading. The possible sources of error with a soil moisture meter is usually due to misreading it or contaminating it with other samples. So to avoid any errors, you must make sure that you wipe the probe before each new sample is taken. So remember, it's wiping the probe, not the meter, so it's got to be the probe. So you wipe the probe between each new sample. Also, you need to insert the probe to the same depth each time to make it valid, and the probe must be properly maintained. So well done if you managed to get that correct and you remember it from third year. If not, then you might need to just watch over the sampling bit again. So the next thing that we're going to move on to is temperature. So temperature can be measured by a thermometer or a temperature probe. So this may be part of another sensor. Make sure that the sensitive bit of the probe of the thermometer is where you want to take the sample, and give it enough time to register and record acclimatise. So you can't take the reading straight away, you have to give it some time to adjust to its new temperature. So there's pictures of all of the thermometers and temperature probes that you might have used in biology. Possible sources of error with a thermometer is placing it in direct sunlight, because obviously it would heat up and wouldn't give you a true representative of what you were trying to measure. Also holding the bulb of the thermometer in your hand, because again that wouldn't give you a true reading, it would give you a reading of the temperature of your hand. And finally, not allowing the thermometer time to acclimatise. You need to make sure that you give the thermometer enough time at the change of condition so that it makes sure that it can pick up on the reading. And also you need to make sure that you repeat the test to make it reliable. The last abiotic factor we're going to look at measuring is pH, and your pH is measured by using a pH meter, which is similar to your soil moisture meter. So your digital pH probe can be pushed into the soil and the results read off of the scale. Some digital pH probes use a solution, so soil samples have to be prepared for testing. Another way you can test pH is by using pH paper or indicator solution and comparing it to the universal indicator scale. So sources of error with the pH meter would be just the same as the soil moisture meter. So it's mainly to do with misreading and contamination. So the probe needs to be wiped off before each new sample. Remember, we always talk about the probe here and not the meter itself. You don't want to wipe the meter, you want to wipe the probe. The probe also must be properly maintained. Well done if you managed to get that correct. If not, then go back and watch different sampling techniques. So pause the video and try to fill out the table before I go over the answers. So we should now know the equipment used for measuring light intensity, soil moisture, pH, temperature, the possible sources of error, and the ways to minimize them. So for light intensity, the equipment and the technique, your light meter would be used, and the meter would be held on the soil surface and pointed in the direction of maximum light intensity, and then the meter would be read. The possible sources of error would be that the observer casts a shadow on the meter or the light intensity changes. To minimize this, for the observer casting a shadow over the meter, you'd make sure that the observer stands to one side of the meter and does not cast a shadow. And for the light intensity changes, you'd make sure that all measurements were taken at near the same time of day as possible. For soil moisture, your equipment used and the way you do it would be that you'd use a moisture meter and you'd push the probe into the soil and read off the meter. A possible source of error would be that moisture remains on the probe. You don't want contamination. So the ways to minimize the error would be to wipe the probe after measurement. For pH, the equipment and technique would be that it would be a pH meter, and again you'd push the probe into the soil and read off the meter. A possible source of error would be that soil remains on the probe, which would contaminate it. So the ways to minimize this error would be to wipe the probe after each measurement. And lastly, temperature. So you can do this by either a thermometer or a temperature probe. If you're using a temperature probe, you need to push the probe into the soil and read it off the meter. So possible sources of error would be not allowing the thermometer to acclimatise and soil remaining on the probe, which would cause contamination. So to reduce this error and to make sure it doesn't happen, you'd have to allow the thermometer time to acclimatise in its new condition, and you need to wipe the probe after each measurement. Well done if you managed to get that correct. If not, then make sure you go over this table and memorise it, because it summarises all of the abiotic factors and how you have possible sources of error and the ways to minimise the error, and that is all common SQA exam questions. So any question on reliability, such as how can we improve the reliability of different types of measurements, anytime you see the word reliable, you need to answer the same way. So reliability questions are answered by saying that you need to repeat the experiment and take an average. So always repeat the experiment and take an average if you get asked about reliability. And you can remember that repeat and reliability start with an R. Now let's look at a line transit. Most of you will have done this in third year. So a line transit can be used to investigate the factors which affect the distribution of organisms in an area where there is an obvious change in habitat, e.g. woodland to grass. So basically it's looking at how abiotic factors are affecting biotic factors. So how would we set up a line transit? We would do this by a piece of string which is pegged out from one area to the other, and samples are measured along it. So the samples will be taken by quadrat for plants and animals, pitfall traps, and then the abiotic factors which affect them. So you'll be looking at soil moisture content, you'll be looking at pH, light intensity, and how these effects are affecting the plants and animals. So usually you take samples each meter from where you start to where you finish. So here's an example of a seashore transit. So samples will be taken at each of these points, so each meter. So you take a quadrat sample, you take a pitfall sample, you take soil readings, so soil moisture readings, pH readings, light intensity readings. So here's a woodland transit. So again, you've got your string, and then at each of these 1 meter intervals, you would take your quadrat sample to find your plants and your pitfall traps to find out your animals, and then you'd also take your soil moisture content, you take your light intensity readings, your temperature readings, and your pH readings to see how it's affecting your organisms. So in this example, you're going from the middle of a field to the edge of the field. So again, you have got 1 meter intervals where you're taking all of these different samples to see how the abiotic factors are affecting the biotic factors. So samples of plants and animals can be taken at each position using quadrats and pitfall traps. Light, moisture, and pH readings can also be measured using light, moisture, and pH meters, and it is possible to relate the distribution of organisms to the changing abiotic factors. So basically, you're linking and finding a relationship between your abiotic and biotic factors. So in an exam, you might get asked about a transit and how it shows a relationship between the abiotic and biotic factors. So let's look at an example of how transits can show the relationship between abiotic and biotic factors. So if we look at this one where we've got a transit from the edge of the field to the middle of the field, and we got these readings. So you've got your sample site, and at each meter a sample has been taken for light intensity, and also quadrat has been done for the abundance of daisies. So we can see at sample site there was 0 daisies and the light intensity was A. So the light intensity, if it's A means it's low, and if it's H means it's the highest. So it goes from A to H. So try to work out what you could write for a conclusion here and for an evaluation before I go over the answer with you. So pause the video and try to write your own conclusion and your own evaluation of these results. So your conclusion should include: what effects did the light have on the distribution of daisies and why this would be? And your evaluation should include: what variables would you have kept the same and what could have made the result more reliable? So the answer to this would be that as the light intensity increases from A to H, the number of daisies increases from 0 to 20. This is because more daisies can grow because they can photosynthesize. The evaluation would be that the time of day would have to be the same light intensity meter would need to be used, the same flower would need to be counted, the soil moisture would need to be the same, the pH would need to be the same, and the temperature would need to be the same. What could have been done to make this more reliable? Repeating the transit and taking an average, just to make sure that we get rid of any atypical results. The next thing we need to learn about is indicator species. So exploitation is when something is used in order to gain benefit. For example, humans have used trees to make paper. The increase in human population has led to overexploitation, taking too much from the environment. Overexploitation has led to a decrease in biodiversity. So examples of overexploitation from humans include overhunting, overfishing, overgrazing, habitat destruction. So for example, desertification and deforestation. Introducing of foreign species and pollution. So air, land, fresh water and seawater can become polluted and biodiversity can decrease. Organisms whose presence or absence that gives information about the levels of pollution are called indicator species, and you need to know the definition of indicator species and you need to know examples. So for example, we have lichens which are sensitive to sulfur dioxide. So make sure you learn the definition of indicator species and write it out many times. So indicator species are species that by their presence or absence indicate environmental quality levels of pollution. So let's look at this in a bit more detail. So let's look at lichen first. So lichen are air pollution indicators. So lichens are indicators of air pollution since different types of lichen have different sensitivity to sulfur dioxide gas. So there are many different types of lichens. So you get hairy, shrubby, leafy, and crusty lichen. So in areas of low pollution, mainly hairy lichen would be found. So a hairy lichen would be where there's low sulfur dioxide and it would be low pollution. Whereas in areas of high pollution, mainly crusty lichen would be found. So that means that there's high concentrations of sulfur dioxide. So as you can see from this graph, when there is a low sulfur dioxide concentration, there is only hairy lichen, so that would be where there's low pollution. Then some shrubby lichen will appear, and then some leafy, and then crusty lichen will appear. So crusty lichen are going to be where there is a high sulfur dioxide concentration, so where it's highly polluted, and hairy are only going to be found where there's low sulfur dioxide, so low pollution. Another indicator species that you need to know about is freshwater invertebrates. Some freshwater invertebrates are only able to live in water which contains a lot of oxygen. Others can survive in water which contains little or no oxygen. So these freshwater invertebrates can show whether the water is polluted or not. So whether you'd be able to drink the water or whether you should avoid drinking the water. So small invertebrates that live in freshwater have particular levels of pollution they can handle. By knowing what the tolerance of particular organisms are and looking at what you find, you can decide on the water quality. For example, stonefly nymph and mayfly nymph live in water that has low pollution and where there's high oxygen, whereas bloodworms and rat-tailed maggots live in polluted water where there's less oxygen. You can remember this because the ones that sound like fairies, like mayfly nymph and stonefly nymph, they are where it's got lots of oxygen and there's low pollution, whereas the ones that have horrible names like bloodworm and rat-tailed maggot, they live in where it's highly populated and there's not a lot of oxygen. So in this graph you have untreated waste being released, so obviously that's going to increase bacteria, which is going to decrease oxygen, and the water is obviously going to be highly polluted at this part here, and then it's going to start getting less polluted and get more oxygen the further that you go downstream. So the biodiversity is going to decrease when there's less oxygen, and you're going to get your rat-tailed maggot and your sludge worm, whereas when the oxygen level starts to increase and you've got your low pollution, you're going to get your mayfly nymph. So this is just saying that with what we've already seen and giving you an example so that you could explain it. So let's go over the different invertebrates and where they're found. So try to answer it before I go over it with you. So pause the video for each of them and try to answer whether they're in polluted or unpolluted water. So your stonefly nymph, would that be found in polluted or unpolluted water? Remember, it sounds like a fairy, so it'd be in unpolluted water. So well done if you managed to get that correct. What about your waterlet? Kind of disgusting name. So it is found in polluted water. Your rat-tailed maggot, again quite a disgusting name. So that is found in very polluted water. So well done if you got that correct. What about your caddis fly larvae? That is found in unpolluted water. Your sludge worm, again disgusting name. So that would be found in very polluted water. Your bloodworm, again a disgusting name. So it is again found in polluted water. Your mayfly nymph, which sounds like a fairy, that's going to be found in your unpolluted water. So well done if you managed to get all of them correct. Now you can complete the activities below to review your learning. So you can complete the questions on page 174 of the National Biofumes Torrance textbook. You can complete the Life on Earth exam practice questions, page 61 to 65. You can also answer the key area past paper questions on distribution of organisms. So we have now come to the end of the lesson, so you should be able to tell the difference between abiotic factors and biotic factors. You should be able to write down about how abiotic factors affect biodiversity. You should be able to create a table to summarise how you measure abiotic factors, which includes the possible sources of error and how you minimise them. You should be able to write down what a transit is and describe how it can be used to show that how abiotic factors affect biotic factors. And finally, you should be able to explain what an indicator species is and give some examples. Well done if you can confidently do all of that. If you can't, then you'll need to watch the video again. That is the end of the video tutorial. Woohoo! Thank you for listening.